Hello, my name is Josca Schoonejans, and I'm currently an Imperial College Research Fellow at Imperial College London. But this work was largely undertaken during my PhD at the University of Cambridge in the lab of Professor Susan Ozanne. So I'm here to tell you today about our work that was recently published in the Journal of Physiology. If a woman is living with obesity, she is more likely to develop diabetes during her pregnancy. Now, children born to mothers with obesity or gestational diabetes have an increased risk of developing obesity, diabetes, and heart disease themselves later in life. Therefore, research is ongoing into suitable treatments that can be given during pregnancy to prevent this. Now, metformin is the most common oral medication to treat gestational diabetes. Since metformin readily crosses the placenta, it can reach the baby and potentially affect its development in the womb. Therefore, we are very interested in studying the effects of maternal metformin treatment during obese pregnancy on both the mother and the offspring. So we do this using a mouse model of obese diabetes-like pregnancy. So we compare offspring that are born to thin mice to those from mums who were given a high-fat, high-sugar diet before and during pregnancy. We also include a third group of obese mice that was given metformin during the pregnancy. Now, in this model, we have previously shown that metformin treatment in obese mums has positive effects on maternal physiology, including her body weight and glucose control. But in this study, we are looking at the offspring. So we followed up male and female offspring of all three groups until they were 12 months of age, which in mice is the equivalent of what middle age would be in a human. So we found that female offspring of obese mums had thicker blood vessels in middle age, and also had higher blood pressure throughout their life when compared to daughters of lean mice. Now, male offspring of obese mums did not show abnormalities in their blood vessels, but instead their hearts showed signs of not being able to fill properly with each heartbeat, also called diastolic dysfunction. Now, maternal metformin treatment did not actually prevent the blood vessel changes that were seen in female offspring of obese dames. Moreover, female mice that were exposed to metformin in the womb were fatter and had obesity-induced hypertension as a result. In male offspring, maternal metformin treatment caused blood vessel hyperreactivity, which is a risk factor for high blood pressure. The hearts of these offspring were also even stiffer than sons of untreated obese mothers, meaning that these mice are more likely to develop heart failure. So we conclude that metformin treatment did not prevent the adverse effects of maternal obesity in either male or female offspring. Moreover, the intervention introduced independent, different cardiovascular problems in exposed offspring, which again were different between males and females. Therefore, metformin treatment had adverse effects on offspring health, despite being beneficial for the mum. Now, this highlights how important it is to study both male and female children in human studies to test longer-term effects of medications used in pregnancy, including metformin. Thank you for taking the time to learn more about our study published in the Journal of Physiology. The full paper was published in Volume 603, Issue 17. Please contact me, Josca Schoonejans, at josca.schoonejans11@imperial.ac.uk if you have any questions.